Here you see what you can do with enzymes and downstream processing in modification on glycolipids. Again, I show you the MEL and here the CL compounds. If you treat a normal fermentation from a MEL-producing strain, you can basically collapse the MEL structures by lipase treatment to MEL-D and MEL-X variants, which is the deacylation, deacetylation by the lipase. So what you do here, you basically move to a higher polarity of the MELs in this experiment. If you do the same for the cellulose lipids, interestingly, you see the opposite. Here we suggest that this is basically a trans-esterification reaction where you get this free fatty acid part acetylated somehow. If you treat these DLs with alkaline, you get a deacylation and the polarity is increasing strongly. You can see actually the differences in foam production or emulsifying properties here. This is a native CL and this is the alkali treated CL where you see the emulsified solution which is cleared much slower than in this case. Okay. Several completely new glycolipids were produced from enzymatic synthesis. They were all done via lipase catalyzed trans-esterification, the next ones at least. And here amino sugars were fused with fatty acids or esters of fatty acids. This is usually a lipase very closely related to novozyme 435. And you do that, of course, in a water-free solvent. The structures here were elucidated with NMR, HPLC was used to purify them, and the reaction parameters were tested and optimized. From these compounds, we received sub-gram amounts, so they were not gone into testing in this part of the project so far, but there are definitely new structures with interesting properties. We also had the enzymatic synthesis of novel terpenoid sugar esters. Here we actually developed a solvent-based and a solvent-free process for novel, not yet characterized biosurfactants, and again determined the reaction kinetics, made actually a recovery process for the substrates and the biocatalyst, again a lipase. The products were analytically validated and tested for their surfactant properties. At higher pH, these compounds have a problem with hydrolysis. At lower pH, they are quite stable. And here you see the general reaction scheme. In a solvent-free esterification, you add basically the sugars or derivatives thereof with the terpenoids. You can react them with a lipase, that is an immobilized enzyme, that you can then filtrate off using vacuum filtration, and after washing, reuse. Liquid extraction gets you the product and the not reacted educts. The sugar will be taken off here. The terpene then can be distilled off and reused for additional reactions together with the sugar. And after chromatographic purification and rotary evaporation, you have the pure terpene sugar ester, and that is possible in the gram amounts. And these compounds were also tested, which I'll show you later. The last interesting enzyme in this part is a glycoceramidase that was engineered for synthesis. Here you do not only use monomeric sugars, but or dimeric sugars, in this case cellulose derivatives, a 2-chloro-4-nitrophenyl cellobiocide for the fusion with a either hydroxypentane 2-one or diols with a different N length here. So here you can actually use this modified enzyme to perform a transglycosylation reaction, which results in cellulose lipids with a yield of about 60%. Structure is shown here with the acceptor 5-hydroxypentane 2-one. Okay, the compounds I've shown you were largely sent to testing, to EcoVer. Here, the testing of the generated biosurfactants for basic physicochemical properties was done. We also tested for some of them for antimicrobial properties, and several of these were produced in up to 100 gram scale to go for full application tests. Okay, here you see one chart of several compounds that went into these testings. On the bottom, the blue parts, the sophorolipids, are already part of products from EcoVer, so you here see them as a comparison. Surface tension and contact angel are typical measurements for surfactant properties. And here we have 4 different terpenols, which I cannot disclose any further, which show surface tension values that are quite similar here. The manosyl erythroid lipids or the cellobeose lipids are also in the same range. What you see that the contact angel here is also in the similar range. A problem here is the CMCs, and here the slow water solubility of these 2 compounds. So here we have the respective testings, which suggest that at least they do have some properties that are good for biosurfactants. The water solubility here has to be improved, and here we have a problem with pH stability, as I've shown you before. Okay, work package 5, we looked at the metabolic pathways that are required to produce biosurfactants. And this was done mainly on pseudozyma strains. Here we investigated one of the strains that showed the most promising profile of biosurfactant amount and composition on a genome and transcriptome level using next generation sequencing. So the genome was sequenced and compared to the genomes in the database. About 1/3 of these genes are highly homologous to related species. And we also did a transcriptome of this. If you look on the genome in more detail, we see a cluster that is known to be required for MEL synthesis in Ystylagomyides. And if you do the transcriptome in the presence of hydrophobic C sources, then you see that about 13 metabolic groups are upregulated, including the MEL pathway. So what you see here is a difference between a glucose field and a mainly soybean feed, where all the genes that are relevant for MEL biosynthesis are upregulated. So by that we identified the respective pathway in Pseudozyma aphides as also relevant like in other strains. Another interesting way to identify new biosurfactant pathways was developed by Selecta. They attempted to use large cosmid shuttle plasmids that are able to go into E. coli and Bacillus species for the integration of large pieces of genomic DNA. So they developed the backbone here of this shuttle. They developed a protocol for highly efficient transformation of these cosmid libraries in screening hosts like Bacillus subtilis. As a control, they used a surfactant-deficient Bacillus subtilis. Surfactant is a known biosurfactant. And they built a control library that should contain this pathway. And indeed, if you transform this into this Bacillus subtilis, the production of surfactant was confirmed by emulsification tests using different screening assays. They were also used for screening of larger libraries, but until now, in this part, no additional positive hits were found. Nevertheless, this technology was considered as highly interesting also for other pathways by the company. In the last technical part, we have focused on bioprocess engineering and downstream processing. Here the focus is of course on cleaning of these surfactants and making them available for products. Two different sets were here investigated, a batch-wise downstream processing and an ISPR-based way. What you see here is basically the downstream processing of MEL and CL that was done batch-wise. So this comes out of a fermentation after cell separation. We have about 60 to 80% purity after recovery of the samples, and the things look brownish. If you want to put them into products, the purity should be more than 90%. And to do that, several different strategies were used. So precipitation of the biosurfactants by acid, reducing charge and so on, but this didn't work that much. Extraction with supercritical carbon dioxide and membrane filtration with different membranes and solvent product mixtures. What you see here is basically the result. For the melts, we could identify membranes and solvent mixtures that lead in a two-step filtration to a purity of about 90%. So basically here you see the melts and the concentrate, second concentrate, and the separated fatty acids. And for the CLs, we even found a better procedure. Here the CLs went almost to 100%. Well, that's barely readable here, but using a membrane hexane mixture, we could basically completely separate the lipids that were in the permeate from the biosurfactant that was in the concentrate. So this was a very successful downstream process that can be used further on. For the sophorolipids, an ISPR process was developed. Focus is here on the cell separation. Sophorolipids occur as precipitates, but also in the soluble fraction of the fermentation broth. And here you see basically two membranes giving different results for the soluble or the sediment part of the sophorolipids, with a total recovery here of over 80% in this in situ process. That was done on a smaller scale. On a larger scale, this is currently still in process. This is a 300-liter fermenter to which a membrane stack is connected, containing these membranes. The first trial was just before Christmas. Currently, they are more facing the technical difficulties with the fermentation at this point, but we are still here in the process, hoping that this week or next week we have the final results from the upscale of this downstream processing. That is for the sophorolipids. Okay, a little bit to dissemination and exploitation. What did we do to promote the project? We have been at several different trade fairs that started with the forum Industrial Biotechnology, the Biotechnica in 2011. We have been at the Hannover Messe and at the Grüne Woche in Berlin, even just in January this year. What we also did, we set up the first international workshop on biosurfactants. That was a workshop at the DECHEMA in Frankfurt last year in May, and organized by the major players in the Biosurf Consortium, Christoph Sildat, Rudolf Hausmann and myself. And you can actually have a look at all the talks and presentations under this link. This brings me to my summary. I hope that I could show you that we produced several new modified biosurfactants. The SLs I couldn't show, but MALs and CLs I could show. The novel biosurfactants were identified and produced from new strains like the trehalose lipids or MEL-X as a new modified surfactant. We had a set of novel biosurfactants synthesized enzymatically, especially the terpenols, but also aminoglycosides and cellulobiose lipids. Some of the biosynthetic pathways, especially for MEL production and their regulation, was identified using next generation sequencing. And we did for most of these compounds that were available in gram amounts application tests and qualified their properties for biosurfactants. Here we still have compounds in testing, and of course EcoVare as a company is always happy to test further additional compounds we will still send to them. The novel downstream processes for the three main glycolipids I've shown you, SL, MAL and CL. This is also still ongoing, and EcoVare already is in the calculation process of the cost reduction that they have estimated from the small-scale experiments by this new ISPR process. With that, I'm at the end. I hope that I could show you that we could go from identification and preparation of surfactants via bioprocess engineering and downstream processing to at least the start of new product development for biosurfactants as a product in the everyday needs we have. So thank you very much for your attention, and I'm open to questions.
Thank you. Do we have any questions?
Yeah.
Right, microphone coming.
So for the most promising biosurfactants that you have, what kind of properties would they finally have? Can you tell us?
So for the sulfurolipids, there's a few new derivatives which will be directly placed into the product collection of EcoVare. I couldn't show that so far. We have MAL characterized very far. There we are still in the final decision rate if, for example, the more hydrophilic ones are able to go into a cleaning agent. But it seems that they are much more promising as emulsifiers, which then would go more into cosmetics or into even food applications, things like that. That would be a different field of application which would not be covered by the companies in the consortium actually. Mm-hmm.
Any other questions? I'll ask a question. I mean, sometimes I hear that with industrial biotechnology products that it's not good enough for them just to be equivalent. They have to often have additional qualities or have better functionality than existing, petroleum-derived products. Do you think that do you think that's the case, or do you think with uses in something like a sort of a green washing powder, you can still get away with equivalence?
Well, actually, the glycolipids have in some aspects, better properties than the APGs, for example, the most common surfactant used. But they're still more expensive.
Right.
Nevertheless, at least EcoVare gets away with selling them as green compounds, also with a green marketing strategy.
Right.
That's very clear. Also, if you go to high-priced products like cosmetics, for example, there is already at least one Japanese company that sells MELs or derivatives of MELs as ingredients for cosmetics. That seems to be working quite well for if you go really to bulk products that are used in industrial applications like biopolymers, you should get very close to the price that is also common for petro-based products. But of course, it's the start of a development. The petro-based industry took 50 years to get to this price. So for the bio-based compounds, biotechnology, it might be faster, but it takes some time, of course, to be more efficient. This is still a discovery phase. Mm-hmm.
Where's the major sort of cost in the process, do you think? Is it the downstream processing or the scale-up?
Well, the downstream processing is certainly one, but the other one is if you have fermentations that take a long time, the time space yield is not good enough. So for glycolipids, you have to ferment them between 1 and 2 weeks. Okay, and that makes, of course, a long time. And even the current products are fermented for 1 to 2 weeks, which are already on the market. So that makes the cost also getting bigger.
So that might be your next project, quicker fermentations.
Yes, or in situ product removal that could actually make you a continuous fermentation with a continuous stream of
fermentation.
products. Yeah.
Okay. With pre-finishing. Okay, any other question? Oh, we've got a question here. Microphone's coming.
I'm interested in your shuttle cosmate that Selecta developed. You say it can be used in E. coli and in Bacillus. I'm wondering, just curious, if the same library would be is expressed in the two strains, do you find different hits?
Well, I can't tell you exactly because I didn't do it. I only know that it works in both, in E. coli strains and at least the company-owned Bacillus strain, which is a patented strain with some specialities, which I don't know too much about. And you find the same hits. I don't know if you find also differences. That was not disclosed.
Thank you.
Okay, if there's no more questions, just thank our speaker. Thank you.
Thank you very much.